This conference will now be recorded.
Okay, ma'am. Now you can go on, ma'am.
Okay. Good evening, everyone. The last class, some network problem issue is there. We stopped some points. Okay. Today we will continue that session. Okay. The last class we discussed about the amino acids and different types of amino acids, the structure of amino acid, and that all we discussed. Okay. What are the types of isomers and that existing in amino acids and different configurations regarding the structure of amino acids. And also here we discussed about the number of amino acids, the structures, configuration, how these are arranged. Okay, and that all we discussed. And also we discussed the different properties of amino acids, that mainly ionization behavior of amino acids, and that mainly existing in Twitter ions and calculation of pKa, and that all we discussed. Okay, here Twitter ions also we discussed, and this comparison of amino acids to simple carboxylic acids and amino acids, this one also completed. Next, calculation of pK values. Okay, histidine has 3 pKa's. Okay, by just counting the first pKa values, we can calculate the pKi. Okay, and here L and D isomers also. Next, coming to the, I think here we stop this. Okay, okay. Here we discussed about all the positively charged amino acids and negatively charged amino acids. And depending upon the charge of the amino acids, here these are existing different ions, cations or anions. And also these are existing at some particular pH, that is called as isoelectric pH. And that contains the neutral charged particles. All are we discussed. Okay, next coming to the absorbance values of proteins. How can we calculate the absorbance values? By using the spectrophotometric technique. Spectrophotometric technique mainly uses the light source. Here we can calculate the quantitative, for the quantitative analysis of amino acids or proteins. We can use the spectrophotometry or calorimetry. It depends on the laws of Beer's law and Lambert's law. Okay, and that mainly causes the. Okay, here the spectrophotometer that mainly works on the intensity of light, here intensity of light and intensity of transmitted light. And these are mainly used for the calculation of the absorbance. Okay, and that is detected by the detector, and that causes the quantitative estimation of amino acids. And here mainly this causes the absorption of UV light. UV light absorption occurs by proteins. Mainly here two amino acids. Two amino acids are mainly responsible for the two amino acids are mainly responsible for the UV light absorption. Okay, what are the two amino acids means here? Tryptophan and tyrosine. These are mainly responsible for the absorption of light. Tryptophan and tyrosine are mainly responsible for the absorption of UV light. Why these are absorbing the light? Anything point? Any observation is there for this? Do you know? Tryptophan and tyrosine. Why these are only absorbing the light? Anyone? Presence of hydroxyl group along with benzene ring in tyrosine. Whereas in tryptophan, tryptophan. The common ring that is present in tyrosine and tryptophan is which one? The common one. Anyone? Presence of indolin in tryptophan only. Whereas in tyrosine and tryptophan, both consist of the common benzene ring. Yeah? And this benzene ring, that is the aromatic side chains of these amino acids, and that are mainly responsible for UV absorption. And also here, we included only two amino acids containing the aromatic ring, whereas the phenylalanine, yeah, because they have aromatic ring, Vidushi Goodma, here phenylalanine also exists in aromatic, and here this also absorbs the UV light, but it absorbs the UV light at some different nanometers, whereas tyrosine and tryptophan, these absorb in the wavelength at 280 nanometers. Here, see the graph here. This is this one. Here, observe this one, this value. Here, 280 nanometers. This is the absorbance maxima of tryptophan and tyrosine. Tyrosine and tryptophan, they absorb maximum near a wavelength of 280 nanometers. Whereas phenylalanine, it absorbs maximum at the 257 nanometers. 257 nanometers. Here, this may be existing this range. Here, 250 range. 257. That is the phenylalanine. Whereas the maximum absorption takes place by tryptophan and tyrosine only. Okay, and this is the absorption of amino acids. Next, coming to the here, in that means here these all amino acids, they contain different structures, and here depending upon the side chains, and these form many types of the bonds, and also showing the absorbing spot. Here also cysteine, it can form disulfide bond. Cysteine can form disulfide bond. By combining the two units of cysteine, it forms a cysteine. That means it forming the disulfide bond. This is the disulfide bond. Okay, this is the disulfide bond. Okay, and that forming the cysteine. Okay, next coming to the here, these are the common amino acids. All are the common amino acids. And some of the amino acids are also exist in nature. And that are called as uncommon amino acids that have many functions are also. These, these are participating in different reactions also. And for these main examples are the 4-hydroxyproline, 5-hydroxylysine, 5-hydroxylysine, 4-hydroxyproline, 6-N-methyllysine, and next gamma carboxyglutamate and desmosine, selenocysteine. These are the uncommon amino acids. We will discuss about these functions in metabolisms, protein metabolisms. Okay, and next here, these amino acids in proteins, they can be reversibly modified. That means the structure of these amino acids can be reversibly modified. And that means here the structural points that are present in proteins, and they can be modified. That forms the different forms, and that are mainly phosphoserine, phospho-3. That means here phosphate groups are added to the serine and threonine and also tyrosine and also arginine. Here methyl groups are added, acetyl groups are added, next glutamate groups. These all are added to the amino acid. That means they can change their side chains by adding the different groups. And that means they can be reversibly modified. Just a minute now. Okay, next here, and also non-protein amino acids are also present, and that non-protein amino acids are mainly sarcosine. These are not, that means these are not participating in protein synthesis. Normally, the replication that continues to form the transcription continues that forms the translation process, and that results in the formation of proteins. Here, proteins are the major constituents of the body, and that means these are also proteins. The amino acids are also forming the different types of protein, and that also producing the non-protein amino acids, and that are of mainly sarcosine, gamma aminobutyric acid, beta alanine, and hr-serine, although diacetylserine, homoserine units, lanthionine, homocysteine, phenylserine, chloramphenicol, cycloserine, and epinephrine, histamine, serotonin, penicillamine, ornithine, citrulline. And these are the non-protein amino acids, but these participating in number of reactions. The main reaction, the protein metabolism that contains the different types of reactions, and that participating in different metabolic pathways, and in that these amino acids are mainly involved. How these are producing means by changing their side chains. The side chains of amino acids are modified, and that forming the non-protein amino acids, and these perform many types of the biological functions, okay, and that are called as the non-protein amino acids. Okay, and next regarding these questions we will discuss later more. Okay, and next today's topic is, we will go for next today's topic. Okay, today we are going to discuss about the classification of proteins and peptide bond formation. I think you know, I think you know about the peptide bond formation, how the peptide bond is formed, how the peptide bond is formed. Peptide bond formation, how it forms. How it forms, ma? Peptide bond. Yeah, by reaction between the carboxyl and amino group by removal of water. Good? Okay. Vidushi, good, ma? Very good. Okay, that means here the peptide is mainly formed in between the two amino acids by removing the water molecules. That means the main bond that is present here in between two amino acids is the C double bond ONH bond. Okay, and that is the peptide bond. Here the two amino acids. That means this is the one amino acid, and okay, this is the one amino acid, and this is the another amino acid. Here these two amino acids, by removing this water molecule is removed, and that causes the peptide bond. That means here two amino acids are needed for the formation of one peptide bond. Yeah, then when two amino acid molecules are linked through peptide bond, then the product is called, when two amino acid molecules are linked through peptide bonds, then that molecule is called two amino acid molecules are involved in peptide bond formation, and that resulting product, this is called as, yeah, dipeptide. Good, ma, Shalini. And that means here single peptide bond is present, but here two amino acids are responsible. That forming the product is called as the dipeptide. Whereas tripeptide means Tripeptide means yeah, 3 amino acids and 2 peptide bonds. Good. Okay. And next, here peptide chains of more than 12. Peptide chains of more than 12 but below 20, that are called as oligopeptides or oligopeptides. Oligopeptides or the peptides and that contain more than 12 and less than 20. Less than 20. Whereas if more than 20 are present, then it is called as the polypeptide. If more than 20 are present, then it is called as the polypeptide. Okay, and here these peptides, sometimes these are also cyclic in nature. Some peptides are also in cyclic in nature. What are the cyclic peptide means? Some decapeptides. That means deca means that containing the 10 amino acid residues and that mainly exist in cyclic in nature. Cyclic in nature. And for these examples are mainly antibiotics, antibiotics that contains a number of peptides and that mainly exist in cyclic. Okay, and for that main examples are gramicidin and tyrosidin. Gramicidin, Gramicidin, gramicidin and tyrosidin. These exist in cyclic in nature. These exist in cyclic in nature. And also depending upon the configuration also here the peptide bonds exist. Some of the peptides they involve both the configurations of D and L amino acid. That is the dextro nature and levo nature. And both type of amino acid that put together that forming the peptides. Okay, that means different types of peptide bonds are also present. And this is a simple structure of a simple peptide. And here, normally the protein sequence that starts with the amino terminal and ends with the carboxy terminal. That means here, first the starting ionic that forms is the NH3 plus. That is called as the amino terminal end. This is called as the amino terminal end. Okay, and next, whereas the ending terminal, that is a terminal end, that is the carboxy terminal end. Okay, and how these are indicated, how these peptides are indicated means here this containing the serine, glycine, tyrosine, alanine, and leucine. And these are the amino acids that are combined by the peptide bonds here. And it is also indicated in short form in the name of the amino acid. Here, serine is indicated in the form of S and glycine G, whereas tyrosine Y and alanine A and leucine A. This is the short form of the structure of the peptide. Okay, and here naming of the peptide, that means it starts at the N terminus, and that means here we can use the full amino acid names, and also here we can use the full amino acid name, and that means depending upon the amino acids here present, and during the combination of amino acid that forming the peptide bonds that are indicated in. And next glycine, tyrosyl, vial, alanine, and last that is the normal name of the amino acid that ends with the normal name of the amino acid. Whereas it can also be indicated in the form of 3-letter code: serine, glycine, tyrosine, alanine, and leucine. And also we can use the 1-letter code. This is a simpler code, and that is the 1-letter code can be used. And this is the naming of the peptides. And here these peptides, these exhibiting different types of functions. And mainly these having the function of hormones and pheromones. What are pheromones? Anyone know? Pheromones. What are pheromones? Hormones means that consists of insulin, oxytocin, and thyroid, and that all contain the amino acids. Okay, and also pheromones. What are pheromones? Pheromones means anyone? Okay, pheromones means these having the attractive nature. These having the attractive nature during the mating behavior. During the mating, here these are attracting the individuals, and that means here the hormones. These are also like the hormones, and that are released during the mating, and that are called as the pheromones. These are the chemicals, and these are secreted by some individual for attracting the other individuals, and that are called as the pheromones. Hormones and pheromones and neuropeptides, and that are mainly, and that means here pain mediators, and that mainly of substance P, antibiotics, polymyxin, biocetracin, different types of antibiotics that are released by bacteria, and also these are existing in protection, and different types of toxins are present in different ones, and mainly these consist of the peptides. Mainly mushrooms they contain the amanitin toxin, whereas scorpions these contains the chlorotoxins, and cone snails contains the conotoxins. Okay, and these are the types of toxins and giving the protection. Okay, and these are the types of functions that are exhibited by different types of peptides. Okay, and here these proteins having the different functions, and these acting as the cofactors and also coenzymes. These acting as cofactors, coenzymes, and also they contain the prosthetic groups. What are prosthetic groups? Prosthetic groups. Prosthetic groups. What are prosthetic, ma? Anyone? Prosthetic groups. Give me response. Do you know or not? Yeah, cofactor coenzyme that is tightly bound to the proteins. Yeah, here these proteins, these acting as the prosthetic groups that are covalently attaching to the cofactors. Okay, and here these are binding to the proteins also. Okay, and here these cofactors are mainly, these are the functional non-amino acid components, metal ions, organic molecules, and these are called as the cofactors, coenzymes. Coenzymes means NAD plus, FAD plus. Okay, and that are existing as the coenzymes, prosthetic groups, other modifications. Okay, and here these are the protein functions. Okay, and next coming to the different types of proteins that are formed by the combination of amino acid and that existing. And here the number of residues and the number of polypeptide chains that differ in these ones. Okay, here main examples for these are the cytochromes, ribonuclease, lysozyme, myoglobin, chymotrypsin, hemoglobin, serum albumin. These are the proteins that containing high molecular weight and also contains the different number of amino acids and number of polypeptide chains. Okay, and depending upon these are present. Here you have to remember mainly myoglobin molecular weight In several times, this one is asked. Molecular weight of myoglobin and also cytochrome C. Myoglobin, cytochrome C, hemoglobin, molecular weight. Okay, serum albumin also. You have to remember these molecular weights. Cytochrome C having the molecular weight of 13,000 that containing the number of residues of 104 existing in polypeptide chain. Single polypeptide chain, whereas myoglobin also single polypeptide chain. Up to myoglobin, this existing single polypeptide chain. You have to remember this table. Okay, read once. Okay, next. And here, these amino acids, these are also classified into two types. Essential amino acids and non-essential amino acids. What are essential? Essential means essential. Cannot synthesize by our body. Good here. Essential amino acids are the amino acids that cannot be synthesized by our body. That means these are to be supplied through our diet. And that are called as the essential amino acids. And the remaining ones are called as the non-essential amino acid. And these are the main essential amino acid. I think you know the order of that sequence. Mat will fly. Mattvillphy. Mat will fly. And that contains the main amino acid isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, valine, arginine, and histidine. And these are the essential amino acids and that ought to be supplied through our diet. These cannot be synthesized by our body. And the remaining amino acids are called as the non-essential amino acids. Okay, some of the peptides has these existing in nature that has a different functions and main dipeptide that is existing here as and it is aspartame. Aspartame is a main dipeptide that acting as the sweetener in food industries. And this also contains the other names that is also called as NutraSweet, Equal, TriSweet, Sadecta. Okay, and whereas the IUPAC name, here this is also called as the NL, that means it is starting with the N terminal, N amino terminal, and here L alpha aspartyl L phenyl, L amino acids, these are the alpha aspartyl phenylalanine 1 methyl ester. This is the structure, abbreviated structure of the this aspartame. Okay, what is meant by IUPAC? IUPAC name means IUPAC name means chemistry, organic chemistry. All are coming from IUPAC. What it denotes? IUPAC means IUPAC means yeah, International Unit of Pure and Applied Chemistry. Good, good. That means here the naming of different. Yeah, good ma. Shalini. Okay. Here these are named by IUPAC. This is the standardized ones. And here these are named as aspartyl phenylalanine 1 methyl ester. Okay. Okay, and next insulin. It is the hormone, it exists in hormone and it is the smallest protein that contains the 51 amino acids in two chains. It consists two chains and here these are linked by cysteine crosslinks. What is cysteine means? Two cysteine units are combined that forms the cysteine. Disulfide bonds are present. And here the A chain and B chain, these both are linked by cysteine disulfide links. This is the 51 amino acid one, A chain and B chain. Okay, and the first A chain consists of the 21 amino acid and the B chain contains the 30 amino acids, and these are linked by disulfide bonds. Okay, and next, something I have missed. I missed some topic in structure of proteasma in three-dimensional structure, and this is asked in CSIR NET exam this time, and hence I'm just discussing with you. Okay, and this is the three-dimensional structure. Already you know about how this three-dimensional structure was formed. And three-dimensional structure, it is mainly based on the primary structure of amino acid. That means the linear chain of amino acids are mainly responsible for determining the secondary or tertiary or quaternary structure. Okay, and here some scientist, one of the scientists, Christian Anifens, he published a classical landmark work, and that showed that the tertiary structure was determined by primary structure. He explained by some experiment. What is that experiment? That he proved the primary structure containing the linear amino acid, and these linear amino acids are responsible for the making of the tertiary structure. Okay, and here it does not require extensive equipment. Okay, and just by using only two components, by using urea and beta mercaptoethanol, he proved that primary structure is mainly responsible for forming the tertiary structure. That means how this experiment works means here mainly he chose the enzyme ribonuclease for his experiment. Why he chose the ribonuclease means here this enzyme ribonuclease means ribonuclease, that means that hydrolyzes only the RNA, hydrolyzes only RNA, and it is composed of single polypeptide chain with 124 amino acid, with 124 amino acids. Okay, and during the this enzyme it exists in three-dimensional shape. This exists in three-dimensional shape and consisting of the amino acids of 124 amino acids. Okay, and here this tertiary structure it is mainly formed by the four disulfide linkages in the active enzyme. Mainly four sulfide bonds. Already I said the three-dimensional structure is mainly formed by using the disulfide bonds and hydrogen bond. Okay, and here mainly the disulfide linkages are observed in the active enzyme, that causing the three-dimensional shape of the enzyme. Okay, and this enzyme, when it is only properly aligned in three-dimensional state, and that only functions. That means it hydrolyzes the RNA. The function of ribonuclease is hydrolyzing of RNA. And this enzyme, it when it is properly aligned, that only functioning. Okay, and that is the ribonuclease enzyme, and this is the ribonuclease enzyme. He did the experiment by using only two components, and that are of mainly urea and beta mercaptoethanol. And what is the function of urea and what is the function of beta mercaptoethanol, ma? What is the function of urea? Function of urea. Function of urea. If you know the functions, you can understand this experiment. Function of urea and function of beta mercaptoethanol. Yeah, beta mercaptoethanol breaks the disulfide bonds in proteins, whereas urea, good Shalini, What is the function of urea, ma? Urea. Don't know? Anyone? Here, beta-mercaptan means that breaks only disulfide bonds. Here, the major bonds that are responsible for the tertiary structure are the hydrogen bonds and disulfide bonds. That means disulfide bonds are breaking by beta-mercaptan, whereas urea breaking just only hydrogen bonds. Hydrogen bonds. Okay, and here these different chemical components are present in this enzyme, and these chemical components are separated by using the dialysis process, and that mainly separating the different chemicals that are present in. How these are separated means here by adding either of these chemicals to the media. Here, how the chemicals are separated is done by using two components that are the urea and beta-mercaptoethanol. And here, by adding either one of these two chemicals to the surrounding media, it is not removed during dialysis. That means the breaking of that enzyme, it is not possible by using the only one of the chemical, only one of the chemical. And here, could remove either the urea or the beta-mercaptoethanol in any order he choose. That means he did the experiment with either of these. Okay, and the order of using these ones, these urea and beta-mercaptoethanol, that resulted in a different compositions, and by using this he determined that the linear chains are responsible for the tertiary structure. And what is the experiment means here? First experiment, what is happening means here he added both urea and beta mercaptoethanol to a solution of enzyme, that is the ribonuclease enzyme. Both the enzymes are added, that means both bonds are breaking. Okay, and that means here the activity of that enzyme is last. Activity of that enzyme is last. And next, after that, he removed the urea by dialysis process, and also then removed beta mercaptoethanol by dialysis. Okay, and also here activity is recovered 100%. That means here the major bonds that are responsible for tertiary structure are maintained after removing the urea and beta mercaptoethanol. That is the activity is 100% after removing. Okay, whereas in experiment 2, what he observed means here he added also both urea and beta mercaptoethanol to a solution of enzyme. To your solution of enzyme. He only first he removed the beta mercaptoethanol. First he removed the beta mercaptoethanol by dialysis process, and then removed the urea by dialysis. But he observed that only 1% activity is recovered. Why it is means here beta mercaptoethanol, that is the strongest bond forming ones, disulfide bonding forming ones. Hence, due to the removal of these ones, here the activity is less percentage. In less percentage. And this is the estimation, general estimation, that is n equal to 8 square equal to 64. That is a 1 by 64. That means here the recovery rate is in terms of n. Okay, and next in experiment 3, what he done means first he added the beta mercaptoethanol to the solution that is removed by dialysis process. It is removed in the second experiment. Here in the second experiment, what he done? He removed the beta mercaptoethanol, and this beta mercaptoethanol again it is added to the solution. It is again added to the solution, and then he removed the urea by dialysis. And finally, he also removed the beta mercaptoethanol. and the activity is recovered 100%. Activity is recovered 100%. That means here already urea is there, and next here the recovered beta mercaptoethanol is adding to that one. Okay, and that causes the activity of 100%. Activity of 100%, and that is the experiment, and that done by amiphanes. Understood ma? This experiment, this is the most experiment, and that is mainly used for the determination of tertiary structure, and that is mainly coming from the linear structure of amino acid by using only two components, that is the urea and beta mercaptoethanol, because these are breaking the bonds that are responsible for the three-dimensional conformation of protein, and beta mercaptomethanol, and that causing the breaking of disulfide bonds, whereas the urea is responsible for the breaking of the hydrogen bond. And here by using these ones he observed, by using, okay ma, I'm explaining that. Okay, here what is happening means, here both urea and beta mercaptoethanol, these are breaking the bonds in three-dimensional structure of protein. Here in the first experiment, what he used, the first experiment, both urea and beta mercaptoethanol, these are added to the solution of ribonuclease enzyme. These both are added to the solution of ribonuclease enzyme. And that means that causing the breaking of the bonds. Breaking of the bonds. And that causes the loss of activity of ribonuclease enzyme. That means the activity is lost. Enzyme activity is lost. Here the blue ones. That means here activity means that is the enzyme activity ma. Okay, here, and also after that, in first experiment only, after the addition he recovered the 100%, he lost, the enzyme activity is lost. Okay, and he removed the urea by dialysis process. Removed the urea by dialysis process, and then he removed the beta mercaptoethanol by dialysis process. But the activity is recovered 100%. That means here the bonds that are responsible for breaking, and that means the components that are required for breaking is removed. These are removed. Urea and beta mercaptoethanol are removed. Are removed means that causing the configuration, three-dimensional configuration. That means the activity of that enzyme remains constant. The activity of that particular enzyme remains constant. Whereas in experiment 2 also he added the same one. Both urea and beta mercaptoethanol are added to a solution of enzyme. And here also activity has lost. But first, what happened here? The difference between experiment 1 and experiment 2, here you first remove the beta mercaptoethanol by dialysis process. Beta mercaptoethanol is removed by dialysis process. Then he removed the urea. But the activity is, the recovered activity is less. Why means here the main bonds that are responsible for three-dimensional conformation are beta mercaptoethanol. Okay, after the removal of urea, we can remove the beta mercaptoethanol. Whereas if we first remove the beta mercaptoethanol, and that causes the changing of the total conformation. The changing of total conformation of that particular enzyme. And due to this, the recovered activity is low. Recovered activity is low. Okay, and that is the main here, the main ones here that you have to observe one, that is, okay. Sorry. Next, coming to the, in experiment 3, what is happening means, in experiment 3 he added beta mercaptoethanol to the solution from the experiment 2. That means in the experiment 2, what we did, beta mercaptoethanol is first removed. Then also we removed the urea. Okay, and after the first removal of beta mercaptoethanol, here we have to add the beta mercaptoethanol. We have to add that beta mercaptoethanol, okay, to the solution from experiment 2. Then remove urea by dialysis. Remove urea by dialysis. And also finally, you have to remove beta mercaptoethanol by dialysis. And also activity that is recovered is 100%. That means in these three experiments, what you understand here means, what you have to understand means here, beta mercaptoethanol is mainly responsible for the conformation of the three-dimensional structure. Three-dimensional structure here, it is mainly maintained by using the beta mercaptoethanol. And whereas the urea that is the urea that is responsible for here for that means the hydrogen bonds that are responsible for the conformation of proteins, for the three-dimensional conformation of proteins, and that are broken by urea, and this is not responsible for the activity, that means beta mercaptoethanol removing or addition, that only changing the conformation of protein, Okay, that means here, due to the formation of disulfide bonds, here the three-dimensional conformation are existing, whereas urea is not responsible, hydrogen bonds are not responsible that much of effect of that disulfide bonds. Okay, and only the disulfide bonds are mainly responsible for forming the three-dimensional structural conformation. Disulfide bonds are only responsible here. Just you have to remember that point. That means here, that means that spatial structure. That means here three-dimensional means that exists in space. The spatial structure, it is mainly contributed by disulfide bond. And these disulfide bonds that are only forming the branched chain. Okay, that means here these are formed by using the linear chain of amino acids. Linear chain of amino acids that are combined together, that forming the disulfide bond. Okay, and that means here the tertiary structure is mainly formed by using the linear chain of amino acids. The linear chain of amino acids, when the disulfide bonds are exposed, and that causing the three-dimensional conformation, that you have to remember. Understood, ma? And this experiment is called Anifen's experiment. Anifen's experiment. That means you have to remember that beta mercaptoethanol activity only. Beta mercaptoethanol activity. Here urea is not that much of activity here. Okay. And next here, and coming to the protein classification, different types of classifications of proteins. Okay, and here mainly these are existing structural nature, contractile nature, transport proteins are present, storage proteins are present, hormonal proteins are present, and these proteins and that have the enzymatic in action, and also these are existing in protection. Okay, energy. Okay, and these are the different types of proteins. And the structural proteins are of mainly tendons, cartilage, hair, nails. These all consisting of the proteins. Okay, that are called as the structural proteins. Whereas the contractile proteins, that means contractile nature showing proteins that are existing in muscles. And transport proteins, transport hemoglobin that mainly causes iron transport, albumin. Okay, these are mainly responsible for the transport process. Next, storage. Iron is mainly stored in the form of ferritin. Ferritin levels are to be maintained. Okay, that is a ferritin. It is the storage protein. Hormonal, insulin, growth hormones. These are the hormonal ones. And enzymatic proteins, hydroxylases, and different types of enzymatic ones are there. Protection, immunoglobulins, these are consisting of amino acids that are existing in proteins. Okay, and that's showing the protection. Energy is mainly given by proteins, that is mainly giving the 4.1 kilocalories per gram. Okay, and these are the different types of proteins. And also based on the physical properties, and here these proteins are classified into fibrous protein and globular proteins. Fibrous proteins. What are fibrous means here? These are insoluble in aqueous solution. Insoluble in aqueous solution, elongated molecules consisting of the several coiled polypeptide chains. Here, based on the physical properties, these are classified into fibrous ones. These are insoluble in aqueous solution, elongated molecules consisting of several coiled polypeptide chains. And for these main examples are the collagens. And collagens, these can be converted. What is the property of collagen means? These can be converted into soluble gelatins by boiling and contain large amounts of residues, hydroxylproline, hydroxylysine, and common amino acid residues are present in collagens. Okay, but here no histone or tryptophan are present, and these contain mainly hydroxyproline and hydroxylysine. And these are the major proteins of connective tissues. And next, elastin. Elastins are similar to collagens, but this cannot be converted to gelatins by boiling. And these are the proteins of tendons and arteries. These are mainly present at tendons and arteries region. And next, keratins. Keratins mainly contain large amounts of cysteine units. This mainly contains large amounts of cysteine units, mainly hair, wool, nails. These are the examples of keratin containing portions, and hair is mainly containing 14% cysteine. Okay, and next, globular proteins. What are globular? Here, these are the soluble in aqueous solution, whereas fibrous ones are not soluble in aqueous. Here, these are soluble in aqueous solution, and these exist in spherical or ellipsoidal shape. And for these main examples are albumins, globulins, histones, and protamins. Okay, albumins. Albumins are readily soluble in pure water, and these can be coagulated by heat function, and these are acting as the carriers of hydrophobic molecules. Okay, for these main examples are the serum albumin, egg albumin. Okay, next, globulins. And globulins, these are the insoluble ones, are only slightly soluble in pure water, whereas very soluble in aqueous salt solution, and these can be coagulated by heat. These can be coagulated by, and for these main examples are enzymes and antibodies. Enzymes and antibodies. And next, histones. Histones are the basic proteins. These are basic. Why these are basic means? These contain large amounts of arginine and lysine that are soluble in pure water. Okay, due to the existing nature of arginine and lysine in the histones are called as the basic proteins. And histones, these are mainly participating in chromatin formation, chromosomal chromatin remodeling that contains mainly histone units. Okay, next, protamins. Protamins are also basic proteins that contain large amounts of arginine. Protamins contains origin but not tryptophan or tyrosine. Okay, and these are mainly found in sperm sets of chromosomes. Okay, next, protein hormones. Proteins, these are mainly acting as the messenger molecules and direct the activities of various cells and organs. And for this, main examples are insulin, adrenocorticotropic hormone, growth hormone, and also antibodies. Antibodies, and these existing, they bind to the foreign substances and activate their elimination from the body. Okay, mainly these existing in anti-RH bodies, antibodies in RH factor and the blood groups. Okay, and these are the proteins. that are mainly classified based on the physical properties and also based on the functions also. Okay, okay, and these are the ones. Okay, another classification is also there, and that can be classified into simple proteins, conjugated ones, and derived proteins. What are simple? Already you see these ones: albumins and globulins, histones and proteins, protamines, and these are mainly forming the complex. And derived proteins are forming the and there is a difference in between globin and globulin also. Here globin and that contains the rich in histidine. Globin contains the rich in histidine, and it only combines with the heme portion and that forming the hemoglobin. Next, prolamines, these are soluble in 70 to 80% ethanol and insoluble in water. But these main examples are gliadin of wheat and zein of maize. These are the proteins that are present in wheat and maize that are called as the prolamins. And histones, and these are soluble in water but not in ammonium hydroxide, and these are present mainly in nucleus that are participating in chromatin remodeling. Okay, next, protamines are also existing like histones but present in sperm cells, and these are soluble in ammonium hydroxide. And like that, different proteins are present. Albuminides, also called as sclerose. Sclerose means muscle ones. And here, and that mainly forming that regions, collagen, elastin, keratin. Okay, and that are called as a simple proteins. And the next type is the conjugated proteins. What are conjugation? Conjugated. That means here these are the proteins that are conjugating to the different groups that are called as the prosthetic groups. Prosthetic groups. Okay, that means with the another protein with exception. That means here these proteins are forming with the other ones that are called as the prosthetic groups. That may be nucleic acid, that may be carbohydrate, that may be lipid urines, and these are forming the complex, and that are called as the conjugated proteins. And different types of conjugated proteins are present, and in that mainly nucleoproteins, mucoproteins, glycoproteins, proteolipids, hemoproteins, metalloproteins. And these are the different types of conjugated proteins. These are the different types of conjugated. Here in nucleoproteins, mainly prosthetic group is the nucleic acid, and it exists in large compact, and main examples are the chromatin and ribosomes. Okay, and that are called as the conjugated proteins. Muco portion means muco means that containing the carbohydrate region. That containing the carbohydrate region. Okay, and that are called as the mucoproteins, and these are mainly used for the pregnancy test, human chorionic gonadotropin, and these contain more than 4% carbohydrate, and that are called as the mucoproteins. And next, glycoproteins, lipoproteins, proteolipids. Proteolipids means proteins connected to the lipid portion. Hemoproteins, heme group containing ones. Metalloproteins, that is the metal ionic combining proteins. And these are the different types of the proteins, and that are called as the conjugated proteins. And next, coming to the derived protein, and these derived proteins are mainly primary derived proteins, and secondary derived proteins. Primary derived proteins are mainly called as the denatured proteins. That means proteins. When these are denatured, that giving rise to the primary ones. That means here these are derived from the proteins. Hence these are called as the derived proteins. And these are mainly denatured proteins. That means the denaturing of proteins, the subunits that are present in proteins are mainly peptides. And that means the denaturing of the proteins that giving rise to the different types of peptides, polypeptides, oligopeptides, proteins, peptones. These are the units that are present in proteins. And during the degradation of these proteins that forming the secondary derived proteins and that are of mainly polypeptides, oligopeptides, proteins, peptones. Okay, and these are the derived proteins, and this is the classification of proteins and classification of amino acid and regarding the peptide bond and the structure of proteins. Structure of proteins. Okay, in the next class we will discuss about the different metabolisms, amino acid metabolism, how these are participating in different metabolic reactions and what are the type of reactions that are present in proteins. Okay, okay. Is it okay? Is it clear, ma, for all of you? Vidushi, Shalini, is it clear? Okay.
Okay, ma'am. And students, if you still have any doubts, you can ask, or you can just comment yes. Okay, thank you for responding. So that's all for today. I hope you all enjoyed. And thank you, ma'am.
Okay.
And slowly you can leave the meeting. That's all for today.